Today, we're going to crack open a groundbreaking scientific paper from 1991 that took a wrecking ball to a really simple but deeply held idea about how life itself is built. It's a fantastic story about a puzzle, a huge surprise, and a total 180 in scientific thinking. You know, it all boils down to one of the absolute biggest mysteries in all of biology. How does this thing, this long, floppy string of chemicals we call a protein, reliably fold itself up into a perfect, intricate 3D machine? A machine that can do everything from digesting your food to fighting off viruses. And how does it nail it perfectly every single time?
Okay, so to really get why this paper was such a big deal, we've got to go back and understand the puzzle that had scientists just scratching their heads for decades.
This is what's known, formally, as the protein folding problem. See, scientists knew that the one-dimensional sequence of chemicals, the amino acids, was the blueprint for the final 3D shape. But they couldn't read the blueprint. It was like having a set of instructions written in a language nobody understood. A massive, unsolved challenge. So, what was the leading theory? Well, for a long time, the whole scientific community was pretty much captivated by this one really elegant, super intuitive idea: the perfectly packed machine. And this idea was captured perfectly, just perfectly, in this 1977 quote from the hugely influential biochemist Frederick Richards. He said the inside of a protein fits together with exquisite complementarity, like pieces of a three-dimensional jigsaw puzzle. That one analogy, that picture, it basically shaped the entire field for years. And this just brilliantly lays out the core assumption, right? The theory was that the reason a protein folded into its final stable shape was because of the specific perfect fit between its little amino acid puzzle pieces. Just like a real jigsaw puzzle, the final picture is determined by how the individual pieces lock together. It's simple, it's elegant, and honestly, it makes a lot of sense. But, great science isn't about what feels right. It's about what you can actually prove. So the authors of this 1991 paper designed this super clever test to see if these perfect puzzle pieces were even real. They basically went on a search for molecular best friends. The logic was super straightforward. If the jigsaw theory is correct, then out of all the possible combinations of amino acids, we should find specific pairs that just prefer each other's company, right? Pairs that fit together way better than any others. If the puzzle pieces are specific, we ought to be able to find them. And here's how they looked for them. They took dozens of different proteins and analyzed them in two distinct ways. First, they measured the fit between every possible pair of amino acids by themselves, what they called binary interactions. Then, second, they looked at how that same pair fit into the little pocket created by all the other amino acids surrounding it. So, they ran all the numbers, they do all this analysis, and what did the data show? Well, the answer was a complete and total shock. They found pretty much nothing. A surprising, almost total lack of any preference at all. Okay, now this is where it gets really interesting. Let's look at the actual data. This chart here plots the size of an amino acid pair on the bottom against how tightly they pack on the side. Now, if some pairs were better fits, you'd expect to see their dots floating way above that line. But look, it's an almost perfectly straight line. Big pairs bury a lot of surface, small pairs bury a little, but they all bury the exact same proportion of their surface. No special pairs, none. Now, you might see that one dot that's way off on its own. That's for two cysteine amino acids. And there's a really good reason for that. They can form a strong chemical bond, literally tying themselves together. But that's not a flaw in the data, it's a perfect scientific control. It proves their method could detect a strong preference if one actually existed. And for all the other pairs, nada. And this next bit of data from the paper just hammers the point home. These are histograms, and they show the variety of fit for lots of different pairs. Now, if the jigsaw model were true, you'd expect to see a tall, sharp spike at one specific value, the single perfect fit. But instead, what you see are these wide, flat, messy distributions. There is no single preferred way for these pieces to pack. The verdict from all this data was crystal clear. You just couldn't argue with it. There are no best friends, no special interlocking puzzle pieces. The way amino acids pack together is, well, it's surprisingly generic. This was a huge finding. Think about it. If the specific packing of the puzzle pieces isn't what creates the final shape, then what does? This result just blew the old theory to smithereens and opened the door to a completely new understanding. The authors summed up their entire field-changing conclusion in one powerful, elegant sentence that completely flips the old logic on its head. The fold determines packing. Packing does not determine the fold.
Let's really break that down because this is a big deal. The old myth was that tightly packing specific amino acids was the cause of the fold, like a specific key fitting into a specific lock. The new reality this paper uncovered is that the fold happens for other reasons, and the tight packing is simply a consequence of that. It's less like a key in a lock and way more like sand filling a jar. The sand packs in super densely, sure, but it does so without a single, unique, predetermined arrangement.
And this realization mattered so much. It told scientists to stop looking for specific packing rules that just didn't exist, which massively simplified the problem. It directed their focus toward other, more fundamental forces, like the hydrophobic effect, which is just the powerful tendency of oily parts of the protein to hide from water. And it also finally explained a nagging mystery: why proteins can often have many of their amino acids mutated and still fold up just fine. Well, if the puzzle pieces aren't that specific, it makes total sense that you can swap them out. Ultimately, the jigsaw puzzle was a beautiful, intuitive idea that just happened to be wrong. This paper cleared away a central myth of biochemistry, and it left scientists with a new and honestly a much more profound question to answer. If packing isn't the architect, then what is the master force that truly designs a protein?